Hello to our friends joining us via recording. We are wrapping up lesson number 6 today. So before we started our recording here, we worked together with groups of classmates to figure out the information that goes on this table from your notes. Can you help me out in the chat? What page is this table found on in your notes packet? Where did this table come from? I can't remember what page it was on. Okay, on page 5, perfect. So this table is on page 5 in our lecture notes. This is a compare and contrast table between two of the most important molecules in lesson number 6. So the first molecule that we're talking about in lesson number 6 is DNA. When we talk about DNA, this is something that we talked about in lab. We've mentioned it here in lecture. Where do I find DNA in a cell? What's the location of your cell's DNA? Assuming we're not dividing, right? Yeah, when our DNA is in its normal location, DNA is stored inside the nucleus. That's the function of the nucleus, right? Is to hold your cell's DNA. DNA can't leave the nucleus. That's part of why I have to build RNA, because DNA can't leave the nucleus. RNA can leave the nucleus, DNA cannot. So when I talk about RNA, and I saw this, I liked this on one of my groups, that when I asked where it was found, it says that it starts in the nucleus, and then it leaves the nucleus. Primarily, when you're thinking about the location of RNA, you wouldn't tell me that the only location of it is the nucleus, because most of RNA, when we're thinking about it, is not in there. But a good way to keep track of what goes on in a cell to help you build a protein is to think about how my DNA lives in the nucleus, I turn it into RNA in the nucleus, and then it leaves the nucleus for me to be able to turn it into a protein. So DNA always in the nucleus, RNA made there, and then it leaves. When we talk about DNA versus RNA, the type of sugar that they have is how we get these different letters in their name. I'll ask you guys for the sugar that's in R, because that's a little bit shorter. RNA has which kind of sugar in it? What's the name of RNA sugar? Yeah, RNA sugar is called ribose sugar. So when we talk about RNA, it's called ribonucleic acid. That's what RNA stands for. So the ribo part of its name means that it has ribose sugar in it. When I talk about DNA, it's called deoxyribose sugar. So if I were to write out the whole name of DNA, it's deoxyribonucleic acid. Deoxyribonucleic acid. So the way they get their name, their abbreviation, comes from the types of sugar that they have. Deoxyribose sugar in DNA, deoxyribonucleic acid, ribose sugar in RNA, ribonucleic acid. Hey, when we talk about the nucleotides, or the letters that are in these two types of molecules here, 3 of the letters are the same. In the chat, can you tell me the 3 nucleotides that DNA and RNA have? What are the ones that both of them have? Yeah, so both of them have A's, G's, and C's. So we'll add that to our list, A, G, and C. When I talk about my last nucleotide that each of them have, because they each have 4, what is the name of the one that I only find in DNA? Or what's the letter for only in DNA? Yeah, so T for thymine, that one is only found in DNA. What's the letter that replaces it in RNA? Which one do I have to use instead in RNA? Yeah, in RNA I have to use that U. I have to use uracil. Absolutely, uracil. This is a big tip-off. Remember, we talked about this last time. If you ever see a sequence of letters that has A's, G's, C's, and U's we know we're looking at RNA. If you ever see a sequence that has A's, G's, C's, and T's, we're always looking at DNA. So keep that in mind, the base you're really paying attention to. Do I have T's or do I have U's? Last question on our table here, single-stranded versus double-stranded. Which of these two molecules is single-stranded? Who's only got one strand? Yeah, so RNA is single-stranded, meaning there's only one side to it. When we talk about DNA is double-stranded, which is how we get the word, we use this in lab. We said that DNA is a double helix. DNA is a double helix because it's double-stranded. Hey, DNA being double-stranded, by the way, we have the complementary DNA strand. So we have the original, and then we have the complementary DNA strand. When we talk about a complementary DNA strand, complementary means it is or is not the same. Complementary, it is or is not the same. Yeah, lots of us chiming in the chat here. It is not the same. Remember with complementary, so we'll put not. When we talk about complementary, it means that you can use the sequence of one strand to predict the sequence of the other. In some ways, they're basically a mirror image of each other. So I do have two strands of DNA. I only have one strand when we're talking about RNA. That, by the way, makes RNA a lot less stable. It's not going to stick around in your cell nearly as long as DNA will because it literally falls apart. I did research as an undergraduate student way back in the day, and I did some research with RNA, and it's really hard because when you try to get it out of a cell, it likes to fall apart. So you have to be really careful and do things really fast to try to help your RNA not fall apart. DNA, like, you can leave it on the lab bench and it's totally fine. RNA is like, I'm peacing out, I'm gonna fall apart, won't have me. So it's a lot harder to work with in the lab. By the way, does anyone remember your nerd teacher has a cat named after a nucleotide? Which nucleotide is my cat named after? Do we have a nerd? Yep, I got my cat Adenine. Fun fact about Adenine lately, she has, for whatever reason, started sleeping in our bed again. She's like 10 years old, so like this is weird for her. We haven't had her sleep in our bedroom for a really long time, but we hypothesized that maybe it's because we're leaving the windows open at night now, and she's like, what are all these smells? I need my people. So Adenine has been sleeping in our bedroom lately. Too much information, right? Oh well. All right, complementary. Here's that word back again. And I know I heard this from multiple groups. We could go through and do this whole strand. I probably won't, just because there's better things for us to do with our time. But when I look here, we're doing a complementary DNA strand. Since we're building DNA, which of those special bases am I using? Since I'm building DNA, am I using a U or T? Which one do I use when I build DNA? Yeah, we're going to use those T's, right? So when we talk about complementary DNA, every time in my original sequence I have an A, I'm going to put in a T. Every time in my original sequence that I have a T, I'm going to put in an A. Every time I have a G, we're going to put in a C. And every time I have a C, we're going to put in a G. So we can go through a sequence here. I'm not going to do them all, but do just a few of them here to show you. We can go through on the sequence and start doing things like that, where we pair up the original sequence with its complementary letter, original sequence with the complementary letter, original, complementary, etc. So when you're going through and doing this process of building the complementary DNA, remember that we get to do A's pairing with T's because we're in DNA. When we do the process of transcription, where we're now turning that DNA into RNA, now every time I have an A in the DNA, I actually put a U instead. So instead of it being our original sequence. So here I'm going to put what we had for the DNA. And then below it, we're going to put our RNA here. So here's what we had in the DNA, right? If I ask you to build me complementary DNA, that's what you would start off with. But on this slide, I'm asking you to do transcription. I'm asking you to build me RNA. When we build RNA, this is what the beginning of our sequence would look like. So here's what you would actually build for me right here. This is where you start with, right? And then we keep going from there. But this is what we would start with, emphasis being that my U is very different because we're doing mRNA. So anytime I have a U, I've got RNA. Anytime I have a T, we're talking about DNA. There's a question in the chat about our double helix, asking how the double helix forms in DNA. So it's a question from page 2. I think I probably have a picture of this too. So let me check because that's a good correlation for us to make. We're talking about our double helix. Here's the double helix up close. Remember when we talked about this in our class earlier in the week, I told you do not memorize the structure of these nucleotides. This is what A actually looks like, what the A nucleotide or the C nucleotide, G and T. I promise you I'm not going to ask you to identify these particular nucleotides, but I showed you this to help you see the way that I form those hydrogen bonds, which is our correct answer for that question. We use hydrogen bonds to attach the two halves of the double helix together. Remember that the reason that T pairs with A and the reason that G pairs with C is because of the number of hydrogen bonds they can make. Thymine and adenine both can make 2 hydrogen bonds, only 2, not 3. Guanine and cytosine want to make 3, so they need to pair with each other because they want to have those hydrogen bonds. They want all of their dipoles to be interacting with something. So guanine and cytosine, always going to pair with each other. They're actually going to connect to each other in 3 different places. Adenine and thymine connect with each other in 2 places. So the double helix forms because of those hydrogen bonds. Yeah, so the question in the chat is hydrogen bonds are weaker bonds. That's correct. The reason, though, that to hold together my double helix is because I have so many of them. So I've got hydrogen bonds right here, and right here, and right here. When you have all of these hydrogen bonds that are holding something together, there's strength in numbers, right? So the more of these hydrogen bonds that I put together to hold this helix in its shape, the stronger it becomes. And part of why, when I talked about DNA being more stable as opposed to RNA, part of what makes it stable too is the fact that it just has these 2 strands of DNA together. Since these 2 nucleotides are interacting with each other, there's not a lot of space between them. When I have RNA and it's just by itself, that gives us some trouble with holding things together. Plus our sugar group, remember DNA and RNA have different sugars. RNA sugar is less stable than DNA sugar. So hydrogen bonds are a weaker bond, 100% correct. But if I have a whole bunch of them and they're holding this whole molecule together, that's enough to make DNA not fall apart. Does that answer the question for you, Jacqueline? Okay, perfect. I know we had, I know we had some discussion going on in our groups about how the process of transcription works. So I want to talk a little bit more about how the process works because we didn't really dive into some of the nitty-gritty on that. And then we'll dive into the nitty-gritty of translation today. So I want to pull a picture for you. I added this little billboard sign, but help me out. In your notes, you have this. What page is this picture from in your notes? Sans the billboard. I added the billboard. Do we know what page this one's from? Okay, we're saying page 3. Perfect. Okay, so on page 3, we're talking a little bit about how the process of transcription works. So let me put a note at the top. This is transcription. let's try again. I get to use my mad skills, right? We're going to delete that word, and we're going to try transcription one more time. Remind me in the chat, when I do the process of transcription, I start with one thing and I build something else. What do I start with and what do I build? In transcription, we start with DNA. Remember, DNA holds all the directions we could possibly need to build any protein. I end with mRNA, which is the specific directions for the protein I actually want. So remember, we said this earlier in the week, your DNA has all the directions for every single protein that your cell could ever need. We arrange those directions in things called genes. So genes are specific directions. So the example in our guided lesson, we're talking about the gene for hemoglobin, for example, that protein that red blood cells have a lot of. If we went down farther in our DNA molecule, if we looked over here, we might have a gene for myosin. That's one of the proteins that our muscle cells use. Or if we looked up in front of here, we might see a gene for collagen. Your DNA has tons and tons of genes inside of it. We don't want to express all the genes, and no cell expresses all the genes. They have a specific set that they express. So to help them find the information that they're looking for, to find those specific directions that they're looking for, we have things called the promoter region. The promoter region is where something attaches. Can you guys help me in the chat? What attaches here to my promoter region? There's even a hint on the slide for you. What likes to come to your DNA and attach right here? Yeah, so what likes to attach at the promoter region is an enzyme called RNA polymerase. RNA polymerase. We broke down its name at the end of class on Monday. When we talk about that word polymerase, does anyone happen to remember what polymerase meant? What does a polymerase enzyme do? Any ideas? Yeah, so a polymerase is something that builds polymers. It builds something. That's what a polymerase does. So polymerase means we're building something. And when we talk about RNA polymerase, not a trick question, what does RNA polymerase build? What does its name tell me it's building? Yeah, its name tells me that we're building RNA. RNA polymerase. This is the specific enzyme or the specific protein that helps me to build that mRNA. So mRNA is the molecule that I need to leave the nucleus to go out so I can make a protein that I need. The way that I can build that is using this enzyme RNA polymerase. So RNA polymerase needs to know where in your big, long DNA molecules to attach to find the particular directions that it's looking for, to find the gene. How it knows where to attach is because it's looking for these things called the promoter regions. So think about a promoter region kind of like a billboard. So way back in the day, I don't know how many of us can relate to this. Back when I first started driving, we didn't have smartphones. So it wasn't like you could just do a Google search for the food that you want while you're driving down the highway. You had to watch for billboards to help you know when there was a McDonald's coming up. Or I'm from Oklahoma before I came here to Texas. So in Oklahoma, we have this place called Brahms, best shakes ever. That's one of our favorite road trips foods, our shakes from Brahms. Oh yeah, Jacqueline likes Brahms too. Awesome. Yeah, so if I'm driving down the highway, don't have a smartphone yet because they don't exist, I see a billboard that says, hey, Brahms 2 miles ahead. I know where I can find Brahms. I don't have a smartphone that tells me. I don't have the directions that tell me, but I saw that billboard that helps me to know where Brahms is. That's the way that these promoter regions are. They're a place for RNA polymerase to see, hey, RNA polymerase, here's where you'd find the directions for myosin. Here's where you find the directions for hemoglobin. So RNA polymerase sees that promoter region, it attaches to that promoter region, and then it can go through and make RNA from the message right here. So the function of the promoter region is to give RNA polymerase a sign. This is where you need to attach, because once you attach here, you can find the information you're looking for. So what happens in the process of transcription is RNA polymerase attaches to the promoter region, then it opens up the DNA double helix. Remember, DNA has two pieces, right? It's double-stranded. So we open it up, RNA polymerase opens it up, and then RNA polymerase is going to start reading the sequence of these nucleotides that it finds in here. It reads them one by one. So it goes through and says, for example, okay, this is a T. So I'm building my RNA molecule. I'm going to put in an A. And then we read the next one here. Oh, this is an A. I need to put in a U. Read the next one here. Oh, this is a C. I need to put in a G. So RNA polymerase starts way over here and slowly goes one by one, reading each of these nucleotides in the DNA double helix. As it reads them, it builds the complementary mRNA. So the process of transcription, we turn DNA into RNA. The way that I do that is RNA polymerase reads these bases one at a time and makes a strand of RNA that is complementary to the DNA. Notice with RNA, we talked about this earlier today, and I'm going to emphasize it. Notice with RNA that we have to use those uracils, right? So if my DNA sequence has a T, I can still always add an A. If my DNA sequence has an A, if I was building DNA, I'd put a T up here. But I'm building RNA, so I have to put a uracil in there instead. Then I go down and I have a C in my DNA sequence, so RNA polymerase would add a G. We slowly work our way down the DNA sequence to generate the complementary RNA. Nicole has a question. Go for it.
So when the polymerase splits the DNA, on either side of the DNA, they're already mirroring each other. Does either side make a different? Do you end up with something completely different from each other, depending on which side of it the RNA is mirroring? Does that make sense?
Yes. So I understand your question. So when RNA polymerase makes a strand of RNA, it's always going to use what's called the template strand. So there are two strands of DNA. They're mirror images of each other. RNA polymerase is always going to use the template strand. It's always going to use the same one to make a copy of it. So it's a little outside the scope of our class. If you take general biology, like biology for majors, you would talk more specifically about it. But it has to do with the way that I build my DNA molecules. The only one that RNA polymerase cares about and actually reads is the one that's the template strand. If it did happen to get mixed up for some reason and try to make RNA from the wrong strand, from the coding strand, we would have some problems because they're mirror images. They're not the same as each other. But every time RNA polymerase always reads the same one. So yeah, I mean, that's a great question. If it got mixed up and it read the wrong one, we're in trouble. So you just assume that every time I give you a sequence and I tell you to do transcription on it, assume that I'm giving you the right one. I might be mean, but I'm not that mean, right? I'm not going to give you the wrong sequence and tell you to make me RNA. So assume that I give you that one. Good question, though. Okay, so we make our RNA. Let me see what I've got in here next. Oh, there's that analogy. If you were not here on Monday, we did talk through this analogy. So check that out. I gave you guys the analogy for the process of transcription, kind of like copying things that live in the library. Let's talk about what happens when we leave the library, or what happens when an RNA message leaves the nucleus. Now we're talking about the process of translation. translation. Hey, help me out in the chat. What page do we start talking about translation on in your lesson? Okay, page 7. Perfect. So now we're on to page 7. So translation is the process that a cell uses to take my RNA message, my directions, and turn them into a protein, turn them into what I actually want to build. So the process of translation, we're going from an information molecule, which is my mRNA. It just has directions. We're going from an information molecule into a functional protein. We go from information to something that does a job. We're translating or speaking 2 different languages here. So if you remember when we were talking about RNA, we said that there were 3 different kinds of RNA. There's the RNA that's informational, that's the mRNA. That's what we made in the process of transcription. When we do the process of translation, we use another type of RNA. The other type of RNA we're using here is called tRNA. Now, technically, tRNA stands for, it's called transfer RNA. But if you remember when we're talking about it and when it was in the lesson, I told you to call it translator RNA, because it's the kind of RNA that speaks 2 languages. It speaks the language of RNA, of nucleotides, so it can interpret what those words are. And it also speaks the language of proteins. Remember that proteins are made out of amino acids. Here's the word that we use to describe what amino acids are. Amino acids are the monomers of a protein. They're the building blocks of a protein. So my transfer RNA, or my translator, the tRNA, not only can it speak or can it understand nucleotides, it also can understand or speak amino acids. So it's taking information and turning it into something that can do a job. So the way that tRNA helps us to build proteins is by working with ribosomes. Remember from lab that we said that ribosomes were the small protein building machines? Their job is to help us put together a functional protein. So the way this works is my mRNA molecule, my instructions molecule that I built in the nucleus and I sent out of the nucleus. It's floating around, it bumps into a ribosome, and the ribosome starts to read it or starts to look at the instructions that are there. Now, ribosomes can only read instructions in really small pieces. This word right here, this word codon, that would be an underline, highlight, star word. We need to make sure we know what a codon is. When we talk about a codon, let's see if you guys can help me out in the chat. What are some of the things that you can tell me about codons? What do we know about codons? Yeah, so codons are a set of 3 letters, right? So a codon, it's just 3 letters, only 3 letters. Ribosomes are like early readers. They can't read very big words. They're still trying to figure out how this process of reading works. So when I talk about a ribosome, it's going to read in 3 letters at a time. It's got to read those easy words. So my piece of RNA comes to a ribosome. The ribosome looks at a codon. It looks at a set of 3 letters. And the ribosome says AAA. It reads the sequence in the RNA. And it says, okay, AAA. And it calls out to the environment. And it says, can anyone tell me what AAA means? And it listens to see if any translators know what AAA means. So it listens. I don't have my code table in front of me, but one of my translators comes by, let's see, and says, sorry, I'm looking up really close. Okay, one of my translators comes by and says, hey, AAA, that is lysine. So my ribosome says, sweet. So tRNA comes in, that's attached to a lysine, an amino acid up there. That comes into my ribosome. My ribosome now has a lysine amino acid in it, and it's happy because it's read this set of 3 letters. Then it goes to the next set of 3 letters. Maybe it says UAU. I think that's a stop codon maybe. It says UAU, says, hey tRNAs, does anyone know what that one means? And the tRNA that knows what that one means, perfect, brings in tyrosine. So my next amino acid that I attach to my chain is a tyrosine. I go down, I read the next set of 3. So I work my way through the mRNA molecule, just reading in sets of 3 nucleotides, reading in codons. As I interpret or as I read what this codon means, I can move on to the next codon. When I move on to the next codon, I bring in a new amino acid for that. And as I bring in these amino acids, you can see here on my ribosome, how I start to build a chain of all of those amino acids that were called for in my mRNA molecule. So my ribosome reads it, it sees what those 3 letters are, calls out to the environment, hey, does anyone know what AAA means? And it listens to hear what tRNA molecule can tell it what it means, because that tRNA molecule is going to bring an amino acid with it to help us build that chain. So the question in the chat is, are tRNA molecules always in ribosomes waiting for mRNA? No, they are not. tRNA molecules are always floating around in the cytosol, waiting for mRNA to be read. So tRNA is chilling out, floating around in the soup of the cell. When a ribosome starts reading mRNA, it'll start kind of grasping for those tRNA molecules, if you will. So that's when they would start getting incorporated into the chain. So tRNA and ribosomes are not always attached to each other, but they're floating around everywhere in the cell. So they're going to help each other read. What other questions do we have right now? Or give me a thumbs up. What are we thinking right now about translation? So Jacqueline's asking what the big picture is. I'll get there. Yep. Big picture is our goal is to make an amino acid chain. That's biggest picture. Well, here, let's do this. We just talked about using the ribosome to build our RNA chain right here. Let me show you a special thing. When we're looking at an RNA sequence up here, there are a few places in RNA sequences that are called stop codons. Stop codons. These are a set of 3 letters. So it's a codon that when that sequence is in my mRNA molecule, my ribosome gets there and tries to talk to the tRNA around it and figure out what amino acid it's calling for, but none of my tRNAs speak that language. So none of my tRNAs can tell the ribosome what UGA means, because none of them can interpret that one. If nobody can interpret that one, my ribosome freaks out and just says, okay, I'm done. We're not doing translation anymore. When we hit a stop codon, this is a set of 3 letters that no tRNA molecules can tell you what they mean, because they don't mean anything. They mean we're done. When we hit a stop codon, the process of translation stops. It's over. Any other codon that we hit, my ribosome will try to read it, and it'll use those tRNA molecules to help it read it. But those stop codons are the end of the line. That's the end of the road there. Let me show you what I do with this sequence to help me build my amino acid chain. So let me show you how I get here. You see in your notes something called the genetic code table. Hey, not a trick question. Help me out in the chat. We do or do not need to memorize the genetic code table. We do or do not need to memorize this. Yeah, we absolutely do not. One of my daughter's TV shows, they say, goodness, no. So goodness, no, please don't memorize this table. I promise you, anytime you're using it on a homework assignment or on the exam, I will give you the table. Do not memorize this table. We do need to know how to use this table to help us with the process of translation. So this is the genetic code table. This is basically, if we pretended that we were a ribosome, if we were going to build a protein, we can't talk to tRNAs. They can't help us out on the exam, even though we can will for them to. They're not going to be able to help us. But we can figure out if the tRNAs were talking to us, what they would tell us we needed to add. So the way that I use this table is filled with the codons. It's filled with a set of 3 letters that I'd find in the RNA. So my RNA sequence, I would go through and break it up into codons, break it up into little pieces, and then I would start to interpret those pieces. I closed my page here with the sequence on it. So let me pull that back up. Bear with me here. Because I'm going to put that sequence on my slide so that we can work on it together. Okay, let's pull it up big here. All right, here comes the typing. We got A, U, G, C, C, A, and I'm not going to type them all because that's going to take entirely too long. G, we'll do one more. C, U, A. Okay, so I'm using some of that sequence that you guys have on page 8. I just typed in the first few sets of letters there. So if you're wondering where this came from, it's from page 8 there. This is 4 different codons, 4 sets of 3 letters that are in mRNA strands. So when I look at my first codon here, it's A, U, G. That's my set of 3 letters here. When I look at my table, notice here on the left, it's labeled for the first letter. The first letter of my codon, notice that I have the 4 possible codons over here, U, C, A, or G. Well, my first letter is an A. So I know that the codon I'm looking for is in this A row right here. I've got an A. The second letter in my codon is a U. So I look across the top of the table, see how it says second letter? My second letter is a U. So I know that I'm in this column right here. Now at this point, I've got this row and this column, which tells me I'm in this box right here. If you want to, you can go all the way over to the far side of the table and it tells you the third letter, lists those third letters there for you. Or you can just look at the codons and find it. When I'm looking at my genetic code table, here is AUG, right? AUG. That has attached to it the abbreviation for the amino acid methionine. So when we're doing the process of translation, I'm going from a codon with 3 letters into an abbreviation for an amino acid, methionine. There's my first one. My second codon, my next set of 3 letters is CCA. So again, I'm going to start here on the left. Here's my first letter is a C, so I'm somewhere in this row right here. Go up to the top, my second letter is a C, so I know I'm in this column. We're somewhere in this box. I could go over to the side and find my A to know I'm in the 3rd position, or I can just find it here, CCA. My next amino acid that I'm going to put into my amino acid chain is proline, PRO, proline. So we go through and use these codons, these sets of 3 letters. This table helps us to figure out which amino acid my message says I'm trying to put in. You start on the left with your first letter to figure out what row you're in. Then we go to the top for our second letter to figure out which column we're in. Whatever box they overlap in is where I'm going to find that 3rd letter to help me figure out which amino acid goes into the chain. Let me mention for you A note, when I'm using the genetic code table, these sequences that we see here, the letters on this table are the codons from the mRNA. This might be an underline, highlight, star idea for you. All the letters that you see on this table are the codons that we find in the mRNA. So when you have an mRNA sequence, you can go directly to this table here to figure out which amino acid is being added. We don't have to do anything like change it into a different sequence. We don't have to build something complementary. You just use your mRNA sequence to go straight to the amino acids on this table. So no extra steps there. We go from mRNA straight into amino acids. Gloria asked the question, why methionine is red? The reason methionine is red is because this is always the first amino acid that we put in every single chain. So it has a special name, actually. When we talk about methionine or met, methionine is called the start codon, or more specifically, AUG is the start codon. So every single molecule of mRNA always starts with AUG. We always start with methionine. And then we can go nuts and go anywhere on the table. But every single time, that's how a ribosome knows we're trying to read a message, is it sees those first three letters, AUG. First three letters every time. If you notice on the table up here, I've got some ones that are in bold. We talked a little bit about these ones here, the bold ones called stop codons. When I talk about a stop codon, remember that a stop codon means there are no amino acids that match up with that message. So if we were to hit in our chain here, let's say our next codon, I'm going to add another one over here. Let's say that after we go through and we translate these ones here, suddenly we bump into UGA. We could again use our table. So here's the row for U and the column for G and A, my last letter in that codon. That is a stop codon. Stop is not an abbreviation for an amino acid. Stop means there's no amino acids that match this. Stop the process of translation. So anytime a ribosome bumps into a stop codon in the mRNA sequence, that's the end of the road. I mentioned in the guided lesson for you, like literally my ribosomes fall apart. They can't keep it together anymore when they hit a stop codon. So stop codons, there's no amino acid that incorporates in at that point. Nobody matches. That means we're done with the process of translation. Let me check the question here. Translation is the process of getting mRNA to a protein. Yep, absolutely right. Christina was right. Yep, that's what it is. When I look at this table, I'm going to clear the whiteboard here and type something new here. When we look at this table, there's an important word for you to be familiar with, and that word is degenerate. I want you to underline, highlight, star the word degenerate. What this word degenerate means is that there typically is more than one mRNA sequence that leads me to the right amino acid. So more than one codon will still get me the same amino acid. So a good place on my table to see that my genetic code is degenerate would be right here. Notice how the codon CCU and the codon CCC and the codon CCA and CCG, all of these codons would all lead me to put in the amino acid proline into my amino acid chain. Or if I look down below it, ACU, ACC, ACA, ACG, all of these codons would lead me to put threonine, that amino acid, into my protein chain. I have more than one codon or more than one set of three letters that gives me the same result, that helps me put in the right amino acid. What that means is it gives you a little bit of grace, if you will. If I build my mRNA slightly wrong, I may still put in the correct amino acid, which means I may still build my functional protein. For my friends who have worked on this lesson already, it's this last letter in the codon that I can sometimes change to still get the same amino acid. What was the special name for this last letter in a codon? What's the fun name for that one? Yeah, that last letter in a codon, last letter in a codon is called the wobble base. And this is when, if we were in class together, we would pretend to wobble for you. I do not wobble, for the record. I'm very much not a dancer. But I would tell you that we've got the wobble baby, wobble baby, wobble baby base. So the wobble base is the last letter in a codon. It's not 100% true every time, but a lot of times, if the last letter in the codon is different, a lot of times we're okay. A lot of times we would still put in the correct amino acid to our chain. That's what's called the wobble base. So I said we had, right, a little bit of grace or a little bit of wiggle room, or maybe you can remember a little bit of wobble room. If that last base is different, we're okay. Because a lot of the time I still put in the same amino acid, even if that last one changes. So I have a question in the chat. If the middle letter is different, no, so if the middle letter is different, we do not always get the same result. So check this out. In this column here for proline, we said that if we have C, C, and then any letter, doesn't matter, CC, whatever, gives me proline. If I changed it from CCU to CAU, I still have two of the same letters, but I've changed that middle letter now. I've changed it to an A. Notice now that I add a histidine instead of a proline. Or maybe I change it to CUU instead of CCU. If I change it to U, now I get a leucine. So the only base that I can consistently change and usually have a better chance of getting the same base in there is that last one. Middle one is a no-go. The first one especially is definitely a no-go. So it's really just that last base that I have any chance of still getting the correct amino acid put in. So this word degenerate means more than one codon leads to the correct amino acid. But that codon theoretically is probably just different by the wobble base. It's probably just that, trying to move my box here, sorry. It's probably just that last amino acid, or that last letter that's different, because that last letter is the wobble base. Yeah, so there's a question in your packet on page 9 that Kai is asking about. That the question is, what would happen if the genetic code was not degenerate? Can you guys help me out in the chat? What are some of the things that you can think of that might happen if the code was not degenerate? What might happen if we didn't have that wobble base? Okay, so Nicole's wondering if maybe we'd have some diseases. Yeah, so we could have some disease. So let's think with yeah, so we'll think with the mutations, and then yeah, so Jesse's chiming in that we might get the wrong amino acid. That's kind of the big picture here when we're talking about the code being degenerate. Our goal is to have as many ways as possible to end up with the correct amino acid added into our protein chain. So by having this wobble base, by having more than one way to get to the right answer, if I do that, it increases my chance of building the right protein. It increases my chance of putting the right amino acid into my amino acid chain. So if I didn't have a degenerate code, you change one letter and you're screwed, basically. I'm for sure gonna put in the wrong amino acid. So I have a degenerate code, that's a good thing for me. because it increases the likelihood that I'm going to build the right proteins. If it was a one-shot deal where you absolutely had to get every base right every time, we're more likely to not be able to build the proteins that we want or that we need. What other questions do we have at this point? Yeah, go for it, Jacqueline.
Okay, so I know I've already asked you this question, but now I'm like more confused. So I'm just looking at the homework because it says the genetic code is degenerate. This means that all of the following codons lead to the addition of the same amino acid in a protein except, so you just have to focus on, because I'm not looking at the last letter, I'm not looking at the wobble base, it's the middle letter that's different. So that's why I was confused on, does that still mean it's degenerate if the middle letter is off? Do you understand what I'm saying?
So I think I'm processing what you're saying.
Yeah, it was a homework question, and I got it right because I just chose out of all four the line that had
so generally speaking, if any letter besides the wobble base is changed, that would mean that we are not going to get the same amino acid. That would be an example of us not using the fact that the code is degenerate to help us. What degenerate means is that you can change the wobble base and still get the correct amino acid. If I change something that's not the wobble base, if I change the middle letter, or if I change the first letter, I'm not going to get the correct amino acid. So this idea of the code being degenerate means that I can change the last letter, only the last letter. I can change the last letter and still get the correct amino acid. So in general, when you're thinking about what degenerate means, it means that I can change the last letter and still get the correct amino acid.
Right.
If I change the middle letter, if I change the first letter, I am probably not going to get the correct amino acid.
Okay, I think it's the way it was worded, but it's okay, now I understand. So it says this means that all of the following codons lead to the addition of the same amino acid in a protein except, and that's why it's the one that's different, because it's the middle letter, not the last letter of the codon.
Correct. Yep. If I change the middle letter, that's not going to give me the correct amino acid.
Okay.
The other thing you can do with that question too, I know that I didn't include the genetic code table on that one, but you could pull up your genetic code table too and see that all of those codons, except for one of them, would lead to the same amino acid. So you could do a little bit of detective work, if you will, and go to a genetic code table and do the process of translation. If I flip that middle letter, then I'm not going to get the same amino acid from that.
Okay. Thank you.
What other questions do we have? Or shoot me an emoji with how we're feeling. Doesn't have to be a happy emoji. Shoot me an emoji of how we're feeling right now. Gloria's feeling pretty good. I like that. Awesome. Good. Look at that. I'm seeing lots of generally happy faces. That's good news. Okay, well, here's what I'm going to mention. We are not going to take we're not going to take class time to do this process of translation because we talked about how to do it. Here's my tip off for you though. When you are doing the homework assignment or when you're doing that transcription and translation practice sheet, when I ask you to do translation, make sure you're thinking about things in groups of 3 letters. So literally on your transcription and translation practice sheet, go through and make yourself little lines to help you. Because remember, the process of translation, we do it in codons, we do in groups of 3. So split up your mRNA sequence into codons, because that's going to help you go through and do the process of translation. And again, we use our genetic code table to go through. First letter is the row that it's in, second letter is the column, and then you're inside that box, you can find your third letter. So the process of translation, go through and split your mRNA sequence up into codons, and then use that sequence to help you build your sequence. Sorry, let me see what my note was. Yeah, and Jesse's is making the note, pay attention to what you're translating. Yep, the only thing, so here might be a note to write down for yourself. The only thing the genetic code table works on is mRNA. I can't use a DNA sequence to translate with the genetic code table. We always have to go, so if we're going to build a protein, we always have to start with DNA that we've turned into, DNA always has to turn into RNA. RNA is what I can turn to a protein. So I can't use my genetic code table to interpret DNA. If I give you a DNA sequence, you better turn it into RNA first. Once you've turned it into RNA, then you can turn it into a protein. So we've got to do that extra step. If I happen to give you a DNA sequence and ask you for the protein, make sure you turn it into RNA first, and then you can make it into a protein. So yeah, good note. Make sure we're watching. Did I tell you I gave you RNA, or did I tell you I gave you DNA? I'll mention, by the way, when you go through and do this sequence from your notes, translating this sequence from your notes, that picture that we looked at that showed us the process of translation with the tRNA molecules, this is actually the sequence that's on that page. So if you want to check to see if you're translating things correctly, let me show you what picture it matches with here. At the end, once you've tried to translate it, if you want to check yourself and make sure you translated it right, it actually matches this sequence right here. So this is what you should get when you do that one that's in your notes. If you didn't get this, we can check ourselves and figure out where we went wrong with translation. So that practice sequence there, that came from right here to give you a chance to practice with it. Let me look here. So that brings us to this table. And I know I had said we'll do it in groups, but I think we'll just do it together as a class to help each other out. We've now more thoroughly today talked a lot more about transcription, and we've talked a lot more about translation. So hopefully this table is going to make a little bit more sense for us now that we've talked a little more about these processes. So let's go through and we'll break it down. We'll do it in the chat together here to help each other out. When I do the process of transcription, transcription helps me I start with something that I turn into something else. In transcription, we start with blank and turn it into blank. What am I building in transcription? Yeah, transcription is when I start with my DNA and I turn it into mRNA. That's the goal of transcription. Where do I take my DNA and turn it into mRNA? What place in the cell do I do transcription? Where does transcription happen in a cell? Well, so the promoter region is where I would attach to my DNA. Yeah, the big picture location in the cell where it happens, this happens inside the nucleus. Inside the nucleus. I do need to have a promoter region on my DNA to be able to make it happen. But big picture, where it happens, this happens inside the nucleus. This is where it's got to happen. When I'm doing transcription, I am starting with DNA. I'm turning it into RNA. I want you guys to help me out. Here's the little pencil up above my pointer finger up here. Go ahead and grab that pencil. I want you to circle what things down here we would say that we use in the process of transcription. Can you circle which of these things in the box down here we use to do transcription? Turning DNA into mRNA. help me understand, when we select amino acids, in the process of going from DNA to mRNA, where do we use amino acids? Can anyone help me understand? A couple of us are saying we don't. We don't actually. Yeah. When we talk about amino acids, we call the monomer the small piece that I use to build my polymer that is proteins. So when we're talking about the process of transcription, we're not building proteins yet. We're just taking DNA, turning it into RNA. I don't actually use amino acids to do that. Christina's right. My amino acids I would use when I'm talking about doing the process of translation. So we'll go ahead and add a box around those ones to start. I use amino acids in translation because in translation we're going to end up with a protein. In transcription, we start with DNA, we build RNA or mRNA. So we're going to use mRNA in this process because I build it. To build my mRNA, I use the nucleotides A, U, G, and C. They help me to build mRNA. And the enzyme that does the process of transcription is that enzyme RNA polymerase. So when we talk about transcription, we use the enzyme RNA polymerase that attaches to my DNA at the promoter region. It reads that DNA, puts it into or turns it into these nucleotides, A, U, G, and C, which I'm using to build this molecule of mRNA. So what I use in transcription, mRNA, nucleotides, RNA polymerase. When we go over here, translation, let's fill in translation. In the process of translation, what do I start with and what do I end up with? What does translation start with and what does it end with? Yep, we start with mRNA, right? It's what I made in the process of transcription. I'm turning it into a protein. Proteins, yep, are made out of amino acids. So when I'm talking about translation, I'm ending up with, or I'm going to amino acids. That's what I want to build. So I start with mRNA. I end up with proteins that are made out of amino acids. Where in my cell, let me give you a hint here, what organelle in my cell does translation? Who does translation? Yeah, ribosomes, right? Ribosomes. Ribosomes, and here's a friendly reminder for us too, lab reminder, because that exam's next week, right? The rough ER, the rough endoplasmic reticulum, it is rough because it's covered in ribosomes. So the rough ER is covered in ribosomes. The ribosomes, these are the two organelles inside your cells that make proteins. Your keyword here with the rough ER, if I was looking for that, would be that it's the big organelle that makes proteins. Ribosomes are the small organelle that make proteins. So heads up, it's all about the ribosomes. Sometimes those ribosomes attach to the endoplasmic reticulum, then we call it the rough ER. So the one other thing that I need to circle down here with my stuff that I use, what's the one other thing that we would need to circle here for what I use? Yeah, so those are found, the nucleotides are found in mRNA. It's going to be those ribosomes. Ribosomes make translation happen. What I would say too here, so I'm gonna erase a couple here. I'm gonna pull off nucleotides because instead of using nucleotides in the process of translation, what we're actually connecting to each other, the monomer that I'm using to build my proteins, is amino acids. So we would say that amino acids are used by ribosomes to turn mRNA into a protein. So tRNA brings those amino acids over to the ribosome, helps the ribosome read that mRNA. How do we feel about this table now? Thumbs up. What questions do we have? Okay, Jesse's feeling a lot better. I like that. Here's my daily penguin. Unicorn, like it. I saw that earlier. I didn't comment on it, but I saw the jack-o'-lantern earlier. That's awesome. Okay, well, here's the last thing that I want to mention today. It's kind of the last activity in this lesson for you. Hannah's covering her eyes. The last part of this lesson talks about sickle cell anemia. This is a disease that is caused by an issue with this process of the central dogma. Let me remind you of that terminology. The central dogma. Remember that the central dogma is just the way a cell takes its DNA and builds proteins. That's big picture. What's the central dogma? We take our DNA, we turn it into proteins. We got to use transcription and translation to do that. In sickle cell anemia or sickle cell disease, you have your normal DNA sequence, what it should be, but there's a mutation in the DNA which causes us to go from having a T, a thymine. In an individual with sickle cell anemia, they actually have an A in its place. By putting an A in its place in the DNA, now when I go to do the process of transcription, instead of making the codon GAG, I make the codon GUG. When I do the process of translation with these codons, I get different amino acids. I've got glutamate compared to valine. When we're comparing glutamate to valine, these are different amino acids. They're also very different, they have very different biochemical properties. So if you remember back from our discussion in lesson number 5, when we were talking about our types of amino acids, remember that they have different personality types. Some of them are nonpolar, some of them are polar, some of them are positively charged, some of them are negatively charged. What happens in sickle cell anemia is I put in an amino acid that's from a different personality type, a different group of amino acids. Remember that my personality types on those amino acids were what dictated my tertiary structure. The personality type of my amino acids dictates their tertiary structure. In the chat, remind me, what's tertiary structure? What does that mean? Tertiary structure. Yeah, that's the 3D shape of a protein, right? So let's go back to a slide here. We start off with the normal DNA sequence that I've mutated. My DNA is wrong. So I make the wrong RNA. With the wrong RNA, then I end up putting in the wrong amino acid. When I put in the wrong amino acid, when my amino acid sequence is wrong, which level of protein structure am I talking about here? What level is the amino acid sequence? What level of structure is that in a protein? It's sequence. Yeah, that's its primary structure, right? So I mutate my DNA. I get an issue with the primary structure of my protein. The order of my amino acids is wrong. When I make the order of the amino acids wrong, then the way that those amino acids interact with each other, the alpha helices and the beta sheets, and then the way that those things fold on themselves, all of that gets mixed up. Now my secondary structures and my tertiary structures come together to form my 3D shape. Now those are wrong because I have the wrong personality type in my amino acid chain. So what happens in sickle cell anemia is we end up putting in a hydrophobic amino acid. When we talk about hydrophobic amino acids, what does that mean? Hydrophobic. Yeah, hydrophobic. We don't like water, right? We're afraid of water. So we build these proteins that normally had an amino acid that has no problems with water. Now I put one in that can't stand water. When I put that one in, it's got a space right here that has a non-polar amino acid in it, or it has a hydrophobic area, can't stand water. Well, if I have a bunch of proteins that have this hydrophobic space that can't stand water, what happens is they all group together. Remember those things that we talked about in protein structure? We called it the hydrophobic attractions. Hydrophobic attractions. So what happens is these proteins have a space that doesn't like water. They find a friend who also has a space that doesn't like water, and they end up attaching to each other to force the water out. So a normal protein would be just fine interacting with water. It's going to stay by itself. Normal hemoglobin is all by itself. In sickle cell anemia, because we traded out for an amino acid that doesn't like water, we end up generating these proteins with pockets that don't like water. And those pockets that don't like water get attracted to each other, and we build big, long hemoglobin proteins. Instead of little individual ones, we build these huge ones. And what ends up happening then is these big, long hemoglobin proteins form chains that will stretch out the shape of my red blood cell here. So we end up with something that's called a sickle shape. A sickle is a tool that they would use when they were harvesting wheat. It's kind of like a curved blade. So we get these things called sickled red blood cells, or sickle cell anemia. Compared to a normal red blood cell that's supposed to kind of be a flat disc, we've got a very different shape in a sickled red blood cell. Hey, not a trick question. Sickled cell red blood cells compared to regular ones, do we think they would or would not be able to do their normal job? Are these cells going to be able to do the job? Yeah, they're not going to be able to, right? Goes all the way back to lesson number 1. We said that you had to have the right shape to do the right job. These cells absolutely don't have the right shape. So they're absolutely not doing the right job. And it all comes down to, at the end of the day, how did we get our cells to look so funky like this? The way we got them to look so funky is we messed up all the way down at the primary structure. If the primary structure is wrong, the secondary and tertiary structure will be wrong, which will lead to the quaternary structure being wrong, which ultimately ends up with our cell that's shaped incorrectly. So sickle cell anemia, an example of why we need all those levels of protein structure that you learned about last lesson, excuse me, all of them have to be correct. If they're not, we end up with a disease like sickle cell anemia. That's all I've got to say today. I talked a lot today, right? I'm done talking. What questions do we have? How are we feeling? Emojis. Here, I'll give us a little penguin while we're coming up with our questions. Oh, he's looking a little bit pathetic today. Poor pathetic penguin. He looks the way that we feel, right, at this point in the semester. Yeah, Hannah said she just got to let it all sink in. Yep, for sure. Practice with it and let it sink in, for sure. This poor scraggly penguin, poor guy. Exactly. Yep. So Christina said this is a good example of anatomy and physiology being related to each other. Absolutely. That's why we took our whole first lesson just to say that anatomy and physiology were related. We will see that coming back again and again all semester. Good. Glad, Tierney, that you're feeling a lot better. Definitely takes practice. Definitely make sure you guys check out that transcription and translation practice sheet. I'm not grading it, but it is something to help you practice, and you can always touch base with your classmates, group me, if you have questions about it. The back page of that paper, or page 2 of transcription and translation practice, has a test type question on it. Please read that question carefully. Please practice answering that question, because I promise you 100% you're going to have a question like that on the exam. So read it slowly, answer it, try to figure out what's going on, especially with that thing on the back side of transcription and translation practice. Any other last minute questions before we call it a day? So on the exam, Gloria, yes, I would give you the genetic code table for that particular question. I didn't give you a genetic code table on the transcription and translation practice sheet, but that was to save space. So you would be able to genetic code table for that. Yes, absolutely. All right. Oh yeah, okay, so Kai asked a good question. She said she was working on the third sequence on transcription and translation practice sheet. There is a stop codon not at the very end. Let me put it to the class. What does that mean for us if I've got a stop codon that's in the middle, not at the end? What do we do with that? So Nicole asked if a new strand starts. No, a new strand wouldn't start at that point. Vanessa's right. If I hit a stop codon, whenever I hit a stop codon, the process of translation stops. So that is the end of the line for that translation. Yeah, so Christina, it could mean that we've got an issue with our protein. It could mean that we were supposed to translate 50 more amino acids, but we had an error in our mRNA and we stopped. So we could make an incorrect protein if we hit that stop codon early. The big picture idea is whenever you hit a stop on, that's the end of the line. There are no more amino acids that go in after that point. So it could make an incorrect protein, or it could be that protein is a shorter one and we just need to stop there. But yeah, whenever you hit a stop codon, whether it's at the beginning, the middle, or the end, whenever you hit that, we are done doing the process of translation. Nothing happens after that. Gloria, I don't quite understand your, oh, okay, so you're asking what the amino acid sequence part of that assignment stands for, I think. Yeah, so that is, I'm basically asking you to do the process of translation. So you have a DNA sequence. I asked you to build mRNA from it. Now use that mRNA with the genetic code table to figure out which amino acids we would put in. So what order would those amino acids go in? Yeah, so Kai, to answer your question, after that stop codon, we're done. Yeah, you don't have to do any more translation. That's correct. All right, well, I am going to call it good today. Friendly reminder, you've got a visible body assignment due tomorrow evening. Make sure you're working on that. Make sure you're working on the lab stuff. That lab exam, again, opens up bright and early Monday morning, goes through Tuesday night. Monday at 9:30, we're going to do our general lab exam review stuff. So plan to come Monday at 9:30. Feel free to email me with questions. Keep working on those assignments, including that review assignment, to help you cover some of that lab material from earlier in the week. And again, if you have questions, feel free to reach out. Just make sure to give me about 24 hours in case I don't get to your email fast enough. So good luck with all the stuff you're working on this weekend, and I hope to see a bunch of you guys on Monday morning.